Hello, welcome to the Curiosity Lab. Today we are talking about a breakthrough that sounds like science fiction, but it actually happening right now in labs and clinics around the world. We are entering the era of programmable or medicine. Imagine being able to open the software of life and fix a bug in the code. That is the promise of this CRISPR technology. So, to understand why this is so huge, we have to look at the problem. Our DNA is the blueprint of life, but sometimes nature makes a typo. These errors, from a single letter mutation to missing a chunk of code, cause thousands of genetic diseases. For decades, doctors could only treat the symptoms, they could help you manage the pain or the side effects, but the root cause, DNA itself, was completely out of reach. So scientists did not just give up. Before CRISPR, we had tools like zinc finger nucleases and talents. They were the pioneers, but they had big problems. They were incredibly complex to design, very expensive, and often inefficient. It was like trying to perform surgery with a sledgehammer. The world needed something precise, scalable, and easy to use. So the breakthrough actually came from an unexpected place, the immune system of a bacteria. It turns out bacteria have been fighting off these viruses for billions of years. They use an enzyme called Cas9 as a pair of molecular scissors. Guided by a little molecule of RNA, these scissors find viral DNA and snip it into pieces. Scientists realized they could hijack this natural defense system and use it in the lab to edit human DNA. So, how does it actually work in a cell? Think of it in four steps. First is the recognition. So, we give the Cas9 protein a GPS coordinate, a synthetic guide RNA that tells it exactly where to go in the genome. The Cas9 scissors make a precise cut across both strands of DNA, and then we just want to knock out a faulty gene. The cells tries to patch the DNA, patch the break made by the enzyme, usually make a small error that silences the gene. So what happens is, by the correction method of the cell, we can provide a healthy template. The cell uses that template to knock in the correct sequence, effectively fixing the typo. We have original CRISPR-Cas9, which cuts both the DNA strands, right? So we have base editors, the precision artists, which can actually change the single DNA letter to another without cutting the strand at all. And now we have prime editors, the master characters, which can directly write new genetic information into a specific site. So this is not just theoretical anymore. In 2023, a therapy called Casgevy was approved. So it's the first CRISPR-based cure for sickle cell anemia and beta thalassemia. Doctors collected the patient's cell, edited them with CRISPR to fix the errors, and returned them to the patient. So for the first time, a lifelong debilitating condition has a functional cure. The arsenal is growing fast. While blood disorders were the first win, 35% of academic research is now targeting cancer, 25% is focused on infectious disease. So there are currently over 140 therapeutics in development. Most are in early stages, but the pipeline is moving towards the finish line very fast. The biggest hurdle now is delivery. So, how do we get these molecular scissors into the right cells inside the body? That is the main challenge. So, one of the best allies is lipid nanoparticles, or LNPs for short. These are tiny spheres of fat that protect the CRISPR cargo and help in sneak inside the cell and release the payload, which is this enzyme. So the path forward isn't without obstacles. Obviously, we have to worry about off-target effects, making sure the seizures do not cut in the wrong place. We also face massive ethical questions. And finally, we have to ensure these one-time cures are affordable and accessible to everyone, not just wealthy people. So the secret weapon for the future is artificial intelligence. So AI is being integrated with CRISPR to design better guides, predicted off-target effects before they happen, and even discover brand new Cas proteins in nature. So the number of publications and patents in this space is absolutely exploding, and which is great thing. The world is betting big on this revolution. The CRISPR market is expected to grow from around 4 billion today to over 9 billion by 2030. So North America, Europe are leading the way, but and I think India should also focus on these technologies and try to spend more money on these new technologies. We are at the dawn of programmable medicine. CRISPR is more than a tool, it's a shift in human history. For the first time, we are not just managing symptoms, we are going to the source code of life to correct the errors. So, the future of medicine is being rewritten one letter at a time. Thanks for listening and follow Curiosity Lab.